Hello, guys, and welcome. If I were to summarize the goal of my rotation project in one sentence, my goal is to find out whether eating donuts would cause us cancer in the long term or not. So let's find out. Cancer is all about mutations. Cancer is the result of the accumulation of mutations. Mutations are changes in our DNA sequence that can be caused by external mutagens or agents like smoking or tobacco or IV radiation, or could be caused by internal mutagens like the activity of a certain enzyme, in this case, the ApoBec. Now, it is interesting to know that mutagens do not cause random mutations everywhere. In fact, each mutagen cause a unique non-random pattern of mutations. These are called mutational signatures. Some mutational signatures like SBS4 have a traceable causation, in this case, tobacco smoking. Others, and actually many, including SBS5, which is the focus of our research project, do not have a well-defined traceable cause. It is also important to note that if we were to admit a patient of lung adenocarcinoma and take a sample of the cancer and start analyzing the pattern of mutations, this patient might be exposed to a number of different mutagens, and therefore the pattern of mutations that we see or observe could be attributed to a number of different mutational signatures. In this case, we have a heavy smoker patient, so 70% of the mutations that we are observing are attributable to the SBS4 mutational signature, which again is traceable to tobacco smoking. And therefore, we deem this sample to have high SBS4 mutational SBS4 contribution, as in 70%, and relatively low SBS5 contribution. As we've just mentioned, the causation behind SBS4 is well defined. However, SBS5, which is one of the most common mutational signatures across different forms of cancer, is still to be well defined. However, our lab might just as well have an answer. Our lab is suggesting glycolytic sugar metabolism to be one of the contributing factors to SBS5 mutational signature. This was based on yeast experimentations or experimental data. However, the goal of my rotation project is to analyze human cancer genomics data to assess the validity of this hypothesis, to assess the relationship between SBS5 contribution and glycolytic sugar metabolism. In other words, I'm comparing samples that have high SBS5 contribution and samples that have low SBS5 contribution, and screening whether the expression of the sugar metabolism genes in the high or low samples are different, whether there is a significant relationship between high SBS5 mutational signature and glycolytic sugar metabolism gene expression. Our hypothesis that there is actually in humans a positive, significant correlation or relationship between SBS5 contribution and glycolytic sugar metabolism gene expression, such that if we are to compare high samples with high SBS5 compared to samples with low SBS5, we would find the glycolytic sugar metabolism genes highly expressed. Now the question is, how are we going to test and assess our hypothesis? We will be assessing the validity of our results through a three-step process. The first step of our process is the most important step. We will be answering the following question: which genes show expression levels that are correlated with, associated with the SBS5 contribution? As you can see, this is a two-step or two-part question. Essentially, we need two types of data to answer this correlation question. We need to know first the gene expression data for each of the samples that we have, the cancer samples that we have, which is data that is already provided by the CBio portal, the source of our human cancer genomics data. The second data input is regarding SBS5 contribution. Now, as you may remember with the lung adenocarcinoma patient example, we will be also in this case analyzing the mutational data of 101 cancer patients using a mathematical code called mutational pattern. This code analyzes each of the mutational data samples and give us a label of high or low SBS5 contribution, and essentially gives us a percentage of how much SBS5 is contributing in each sample. In this example, for example, sample, for example, 41% of the mutations that we observe is attributable to SBS5 contribution. Now that we have the two types of data that we need, we will be investigating the linear correlationship between those two data types, such that we will be looking into how is the gene expression, for example, of gene 1 is related to SBS5 contribution across the 101 samples. Is the expression level positively correlated, negatively correlated, or not correlated at all? And essentially, after obtaining those correlation values, we will be ranking them in a list from the most positively correlated to SBS5 to the most negatively correlated to SBS5. In the second step of our process, we will be essentially feeding the list of genes that have been ranked to a method that we call gene set enrichment analysis. A complicated term, I know, but GSEA, or gene set enrichment analysis, is actually one of the most intuitive steps in this entire presentation. What GSEA allows us to do is that it allows for a less complicated way of analyzing the results of thousands of genes. As you can see below, bottom right, you see thousands of genes that are positively correlated to SBS5 in our list. It gets complicated too fast if we try to analyze each individual gene and its corresponding correlation value to SBS5. However, what GSEA does is that it uses a database called Gene Ontology. Gene Ontology is a database where scientists have grouped hundreds of genes under fewer terms that we call Go terms, based on the molecular function and roles of these genes. For example, this Go term denotes for glycolysis genes, and it carries hundreds of genes that are involved in glycolytic sugar metabolism. So what GSEA does, instead of analyzing individually thousands of genes and their corresponding correlation values, it groups the genes under Go terms and give an overall correlation value to the group of genes. But the question is, how does GSEA give an overall correlation value to a group of genes, some of which might have negative correlation value? It goes for a hike. There are two rules for this hike. One is, if the GSEA software finds genes under the Go term in the list, then it goes up. If it does not find genes that are under the Go term but not in the list, then it goes down. If it finds the gene and this gene is positively correlated, like JAK9 in this case, it goes up. If it finds the gene like ENO1, but it is negatively correlated, then it goes down. And it goes through the entire data set. The ones that are shadowed by black are the genes that were found, and accordingly, we go through the hiking experience in its entirety, and the peak of the hiking experience, whether upward or downward, will be the overall correlation score. And essentially, this overall correlation score tells us if in this case, the overall correlation score is positive. So that means that overall, the group of genes are positively correlated to SBS5 contribution. GSEA repeats this exact process for all of the Go terms that we have across the list. And essentially, we will then end up with Go terms and their respective overall correlation scores or enrichment scores. And then we will be mapping our result and visualizing them, as you will see in the upcoming results slides. Now, the first way of visualizing our results is taking the Go terms and their respective enrichment scores and pick the top 10 Go terms. In other words, the top 10 Go terms that are positively correlated to SBS5 contribution. It is worth noting that we get the enrichment scores and we normalize them to be able to cross-compare across different Go terms or gene groups, because each gene group have a different number of genes. So to be able to cross-compare them across them, we need to normalize the enrichment scores. Now, what trend do we see in this result? The trend that we see across the top 10 Go terms is mRNA expression, regulation, transport. But is this related directly to glycolytic sugar metabolism? Maybe, but it is also related potentially to many metabolic pathways. And that's why this result is still not specifically or directly related to glycolytic sugar metabolism. And so we need more specific results. Another possible way of visualizing our results is the tree map. The tree map gives us the chance to see what functions or which functions are getting upregulated in correlation to SBS5 contribution. And so, for example, one of the highlighted results in here is ATP-dependent activity that is getting upregulated in samples of high SBS5 contribution. Now, is glycolytic sugar metabolism in ATP-dependent activity? Of course, because it uses ATP and it also generates ATP. So it influences other ATP-dependent activities. But it is also important to mention that this is not yet also this is also not a specific result that is directly related to glycolytic sugar metabolism. And so ultimately, our current data is one not suffice to fully support or negate a positive, significant correlation between SBS5, mutational signature contribution, and glycolytic sugar metabolism. And so for our future directions, we indeed need to crack through more data sets to have a better representative sample. And also we need to modify the code slightly. Why? We need more specific Go terms. As we've seen previously, the Go terms are not that specific. And also we need to have a look at the bottom 10 Go terms, not only the top 10 Go terms, in order to look at both the upregulated and the downregulated gene sets. The overall significance of our rotation project is that if in fact there is a positive significant correlation between glycolytic sugar metabolism and SBS5, then we can recommend patients with high SBS5 cancer conditions low sugar diets as a therapeutic personalized medicine approach. And I would like to thank all of those that have been involved in this rotation project, including my principal investigator and the current lab members. To answer the question of the night, would eating a donut cause you cancer in the long term? Maybe, but we actually need more data. So until next time, and thank you for your time.